Hi everyone, I'm Xinyi. I'm a graduate student working with Carolina and Shiva. I'd like to present our multi-modal framework for understanding a holistic representation of cell states and tissue organization. So we've heard from previous talks that there are many ways of understanding cell states. Not only can we look at the gene expression to understand transcriptional cell state, we can also look at chromatin images, which contains very rich information of the mechanical state of cells. And we can look at additional modalities, such as protein stains, or how cells organize in tissue by looking at spatial transcriptomics, or looking at tissue imaging. But the challenge coming with all of these data is that it's very hard to compare across modalities because we cannot easily just compare image pixel intensities to the value of gene expression. So in order to fully utilize all of the available data modalities, we need to develop a method that can take into account the very different measurements. And we propose a framework for doing that. So we have one autoencoder for each data modality, and then we can have the autoencoders to jointly encode into the shared representation that now have a union of information from all the input data modalities. With this shared representation, we can then perform downstream analysis such as visualization and clustering, not only comparing the similarity of cells based on gene expression, but also comparing the cells based on if their chromatin image is morphologically similar. So now for example, if we cluster the cells based on the shared representation, then the cells will be grouped together based on their similarity in all of the input modalities. So we then apply this model to look at the spatial transcriptomic data set of Alzheimer's disease mouse model. And now we're integrating into the shared representation the gene expression, the cell chromatin images, and location of cells in tissue. And we can then look at what the clusters tells us and how does it differ from the clustering of a single modality alone. So if we plot the clusters back into where they're located in the mouse brain, we can see that it nicely outlines where the cortex region is and where the hippocampus region is. And it's very consistent across the different mouse brains. And if we look closely into this cortex region of mouse, our clustering is saying that there's this novel separation in the mouse brain cortex, as this differs from the horizontal layers in a reference atlas. So we wondered what's separating this mouse cortex brains. And if we look at an orthogonal stain that we did not actually use to train our model, which is the amyloid beta plaque, a pathological protein associated with Alzheimer's disease, we can see that this novel separation in the cortex region actually corresponds to how likely the plaque is going to accumulate in that particular region. And in particular, a cluster 3 region is more likely to accumulate more plaques and larger plaques. So this tells us that the integration of multiple data modalities is able to identify this spatial progression of Alzheimer's disease and the likelihood of the spatial regions in accumulating the pathological proteins. And since we have the power of multimodality, we can then look at disease markers not only in terms of the gene expression differences, but we can also look at the biomarkers in terms of the difference in cell chromatin. So for example, we can look at where in this chromatin image is more predictive of that there's an amyloid beta plaque nearby. So we indicated this predictiveness of chromatin image of an amyloid beta plaque in the image by the intensity of blue pixels. And it turns out that the predictiveness is not only highlighting particular cell types, but it's picking out very specific single cells within one cell type, for example, this microglia, but not the microglia next to it. It's not only just picking out an entire cell nucleus, but it's also picking out denser regions in the cell nucleus, such as this region in the inhibitory neuron, but not the entire nucleus. So this leads us to the hypothesis that perhaps our model is really looking at the denser regions of the chromatin or the heterochromatin region for the prediction of amyloid beta plaques. So if we quantify the heterochromatin across different cell types, we can see that there's a very consistent increase of heterochromatin, whether it's in neuronal cells or glial cells, when we compare the AD mice to the control mice. So this tells us where chromatin is a very simple and cost-effective biomarker. It contains a lot of information about cell states and can be directly used as a disease biomarker. And because it's so informative, we then wondered, can we use chromatin as a way to predict missing modalities? So as we've heard before, that in the same experiment, we cannot simultaneously measure all the possible modalities. And that's especially the case when we are going into the measurement of proteins. So if we want to understand protein localization, we can take protein stains. However, we have 20,000 different proteins in a human that can be possibly expressed in a human cell. And then if to study this protein localization across all the possible cell lines, then this would be experimentally infeasible. And because to fill up this matrix of protein localization in the different cell lines, we need to perform one experiment for each pair of protein and cell line. So people have actually done that to create one of the largest existing database for protein localization, namely the Human Protein Atlas. And even after many years of efforts and resources, our understanding of protein localization is still incomplete. That we only have 41 different cell lines measured in the HPA, and we only have 17,000 proteins measured in the HPA. And more importantly, not all pairs of these proteins and cell lines are measured, that only 0.25% of all the possible pairings were measured. And this is because the number of proteins that can be measured in the same experiment is limited. So it's perhaps not possible to experimentally profile all the possible pairings. So we need a computational tool that can allow us to predict the protein localization from measurements that we have. We thought that if we combine the protein sequences together with cellular stains that tells us the cell states and cell types, we can then make predictions of the protein localization with a single cell and cell type specificity. So to do that, we first look at the sequence representation of proteins by using a language model. And the ESM2 is a structural prediction model that takes protein sequence to predict protein structures. We tailor the sequence representation learned from ESM2 to the particular task of localization prediction by training this classifier that takes the sequence representation to predict the localization labels. We can then learn cell states from cellular stains, and we're not only using the chromatin stain, but we also have stains for microtubule and ER in the Human Protein Atlas. So with this image representation that tells us the state of single cells, we can then combine the sequence representation with image representation to make a prediction of that particular protein in a single cell. So we tested this model across many unseen proteins and unseen cell lines that we did not use in the training of our model, and we can quantify the prediction errors across different proteins. If we categorize these prediction errors across proteins in different cellular compartments, we can see that our prediction is quite accurate and performs equally well across different compartments. And we collaborated with Fei Chen at the Broad Institute to perform experimental validations to see, in addition to predict localization in unseen proteins and unseen cell lines, can we also predict localizations in new experimental contexts. So we looked at two cell lines that the model has seen in training, and we can see that our model prediction aligns quite well with the experimental validation, even for proteins that were not used in training. And we can then look at cell lines that the model has never seen before. And again, our model performs quite well and aligns well with the experimental validation. And even for a cell line that's not included in HPA. And now since we have the ability to make predictions of proteins and cell lines that the model has never seen before and fill up this matrix of proteins by cell lines to understand protein localization, we can then be able to perform tasks and understand protein properties that were not accessible before because of the missing data. And one such protein property is how protein localization varies across different cell lines. In the Human Protein Atlas, one protein is only measured in up to 3 cell lines, so it's hard to understand the variability of proteins across many different cell lines. And since we filled up this matrix of proteins by cell lines, we can now compute the variability of proteins across many different cell lines. And if we then rank the proteins based on their variability, we can see that the most variable proteins are associated with processes that are known to be dynamic, such as regulation of transcription, cell differentiation, and chromatin regulation. And here's one example of a protein that our model predicted to be highly variable. So for example, in this MCF7 cell line, the protein is more in the inside the nucleus, whereas in a different cell line, like this MB4 cell line, the protein is more outside of the nucleus. And we can also do the same analysis not only on the cell length level, but to also understand the variability of protein localization on a single cell level. And since we're using a multimodal input to make a prediction of protein localization, we started to wonder that which cellular compartment that we used as an input is contributing to this variability of localization. Additionally, we showed that our model can predict quite accurately for many different proteins, but where's the limitation of such a prediction? Are there features in protein localization that cannot be derived from cell states learned from cellular stains? So to answer these two questions, we developed a model that not only integrates different modalities, but also explicitly disentangles what's the shared information between the different modalities and what's the modality-specific information. Because in this case, if we want to know where's the limitation of a cross-modality prediction, we can then look at the shared information versus the modality-specific information, and only the shared information captured by the shared latent space would be something that's predictable in a cross-modality prediction. And then we can also try to interpret what's in the shared space and modality-specific space in order to understand which component in the input modalities is contributing to the variability of protein localization. And because we have a very segmented latent space, that we have a shared latent space versus two modality-specific latent space, in order to make such training possible, we're having a novel training scheme that instead of training the encoders and decoders together as a standard autoencoder, we have a two-step training process that we train a randomly initialized latent space together with a decoder. And then to generalize to cells that were not involved in the first step training, we have a second step training process that trains encoder to map the input modalities to the shared latent spaces. So with this model, we can then disentangle the shared space and the modality-specific space and compare the chromatin stains versus the different protein stains. So for example, if we want to know if we actually need to experimentally measure the protein stain for gamma H2AX versus predicting gamma H2AX from chromatin stains and protein sequence, we can look at the shared space and modality-specific space, and then we found that for some general characteristics of gamma H2AX, such as intensity, that's something that's in the shared latent space, and that can be predicted from chromatin stains. Whereas if we want to understand what's the number of DNA double-stranded breaks marked by the foci counts of gamma H2AX, that's something captured in the protein-specific space, and that has to be experimentally measured and cannot rely on cross-modality prediction from chromatin to predict the gamma H2AX foci counts. Additionally, we've seen that the protein localization are quite variable across cell lines and also across single cells. We want to understand what's the source of this variability. Is it the nucleus stain that's different, or is it the microtubule stain that's different, and which one is causing the differences in the localization difference across different single cells? So to do that, we can probe the shared or modality-specific latent space by clustering the cells in each latent spaces and look at the properties within each cluster. For example, we can look at what's the intranuclear proportion of proteins in cluster 2 versus cluster 1. And if there's a significant difference in intranuclear proportion in a shared space, then this means that the variability in the protein localization within the nucleus is something that's captured by the shared space. So for example, if we do this for the CLNS1A protein, then we can see that the two clusters in the nucleus-specific space show a significant difference in the intranuclear proportion. And specifically, if we look at the high intranuclear proportion cells versus low intranuclear proportion cells, that there's a significant difference in their nucleus morphology, and more specifically, that the cells with high intranuclear proportion of this protein has a higher content of heterochromatin. And we can similarly do this for other proteins such as DDB1, and we found that the variability of DDB1 localization is something that's not related to the nucleus, but is instead captured by both the microtubule and ER. And the variability is associated with the intensity of microtubule and ER channel, as well as the volume of cytoplasm to the ratio of the volume of cytoplasm to the nucleus. So to summarize, I've shown that our framework not only learns a right representation that takes into account all different modalities and allows us insights into the cell state and tissue organization differences beyond what we can learn from a single modality, we can further disentangle the contribution from different modalities and learn the shared and modality-specific latent spaces. And this not only allows us to have a better experimental design in terms of understanding the limits of cross-modality prediction, but also allows us to understand the contribution of different cellular compartments to an observed phenotype. And with that, I'd like to thank my mentors and collaborators, Caroline, Shiva, and other group members. Thanks.
And I think we have time for one question. Any questions? Or?
A very nice presentation. Just one quick question. I wonder whether the chicken and egg, paradox is that when you try to measure the chromatin structure to predict, the Alzheimer disease state,
Yeah.
and I wonder whether, the chromatin was changed as a result of the disease state, or you use the, chromatin as a predictor for the disease state.
Yeah, that's a question we've been thinking a lot, and that's a question we don't have answers for, and we need additional either time course data or perturbation data to understand if the chromatin change happens before the disease change, or if there's something that's upstream of both like cell state change and chromatin change.